Syn of serine. Serine is formed from 3-phosphoglycerate to PEP transamination and phosphatase. Syn of glycine. Glycine is formed from serine via hydrolysis and transfer methyl group to tetrahydrofolate. Syn of proline. Glutamate semialdehyde spontaneously cyclizes to form a proline precursor which is reduced to proline. Syn of arginine. Arginine synthesized sequentially in the intestine epithelial and kidney proximal tubules. Arginase not expressed in these, and arginine is required as dietary supplement. Threonine is the precursor of interconversion of glycine and serine. From threonine, 1 carbon transfer from FH4. Cysteine synthesis. Synthesis of cysteine requires condensation of homocysteine and serine. Sulfur comes from the essential amino acid methionine. Cysteine breakdown. Cysteine breakdown involves oxidation, desulfation and taught to pyruvate. Glutamate. Central role of glutamate in AA biosynthesis. Glutamate carbons to and from glucose in liver. Glutamate carbons from histidine are not from glucose if ornithine is in excess of the amount needed. For urea cycle, it is transaminated to glutamate semialdehyde, which is oxidized to blank are the only ketogenic amino acids: leucine and lysine. Cofactors required for amino conversion: pyridoxyl phosphate, transaminations and other rexins requiring amino transfer, tetrahydrofolate, folate B12 involved in C1 transfers, tetrahydrobiopterin involved in ring hydroxylations. Direct metabolism of amino acids to TCA intermediates. Alanine, glutamate and aspartate to pyruvate, acetoglutarate, oxaloacetate. Threonine. Threonine metabolized to pyruvate, acetyl-CoA and glycine. However, its most common route is dehydratase, which leads to propionyl-CoA then succinyl-CoA. How to make glutamate and aspartate directly from glutamine and asparagine. Glutaminase and asparaginase respectively. Reaction occurs in the liver. What makes glutamate? Alketoglutarate by glutamate dehydrogenase. Proline synthesis. Cyclization and reduction. Serine to glycine RxN components. Blank hydroxymethyltransferase, H4-folate, PLP, BH4 synthesis. From GTP and regenerated from using BH2 reductase to form it from BH2. Side chains in uroporphyrin. Acetate and propionate. Side chains in coproporphyrin. methyl and propionate. Side chains in protoporphyrin 9 and HUMA. Methyl, ethylene, and propionate. Which steps of hemosynthesis occur in mitochondria? One, glycine and succinyl-CoA-2-ALA, rate-limiting step 6, coproporphyrin oxidase and 7. Protoporphyrin oxidase RSNS, 8 protoporphyrin 9 and beta-phe protoporphyrin 9. Causes of jaundice. Lysis of red blood cells, liver damage, or obstruction of bile. Creatinine metabolism. Arginine and glycine to guanidinoacetate to blank to blank.